Hardy-Weinberg Principle. This topic is taken from the chapter of evolution. Okay, so this is very important principle in the point of view of exam, which is Hardy-Weinberg Principle. So the frequency of occurrence of alleles of a gene in a population remains constant through generations unless disturbance such as mutations, non-random mating, etc. are introduced. So this is the Hardy-Weinberg principle. This is very important. So we have gene. We know that we have gene for a height and it has two different alleles, right? Height has two different alleles, that are tall and dwarf are the two different alleles of a gene height. So basically height is a gene and tall and dwarf are the alleles. Okay, so alleles and genes are not same. They basically are not same, right? In any population there are alleles for a gene or there can be a multiple alleles. Okay, so the ability to transfer from one generation to another generation frequencies can be found out. Okay, so there are alleles fixed and genes which transfer from one generation to next generation are also fixed. Hardy-Weinberg in his principle, he simply said that the frequency of occurrence of alleles of a gene is mainly constant. Constant means 1. Okay, it is 1. If the disturbance occurs such as mutations, non-random mating, then only the constant changes. Okay, then otherwise it is always constant means 1. Hardy-Weinberg decided to explain this concept in the mathematical way. So he utilized maths to make this concept very understandable. So let us understand first what is genetic equilibrium. Genetic equilibrium, gene pool remains constant. In the genetic equilibrium, gene pool remains constant. We'll understand what is gene pool and what is genetic equilibrium. So it is a state which provides a baseline to measure the genetic change. Let us understand what is genetic equilibrium and also what is genetic pool. So to understand this concept, let us take one example. So for example, let us say there is a capital A and small a. Okay, and these organisms are diploid. They are diploid organism. Okay, then so let us find what kind of combinations it can create. So basically it can create when it crosses, it can create capital A, right? Capital A and capital A and small a. These are the 4 combinations it can create, right? From these two alleles we can get the 4 combinations. So this much is only possible. So out of these 4 combinations, nothing is possible, right? So this represents the gene pool. This is the gene pool and it will remain always constant. That is 1. It will remain always constant, right? Sum total of all the allelic frequency is 1. Okay, that gene pool always remains constant. So sum total of all the allelic frequency is 1, right? Obviously I have told you right now. Then individual frequencies are represented as P and Q, such as in a diploid where P and Q represent the frequency of allele A and small a. Okay, let us understand this concept. Okay, so let's say there are some alleles capital A and small a. Okay, so there are 2 alleles. So consider the frequency of capital A by P. Okay, these are the frequencies. Consider the frequency of capital A as P. Okay, then the frequency of small a by Q. Okay, so we can simply say it is PQ. So if 2 alleles are same, when the 2 alleles are same, that is small a or capital A, then we call it homozygous. So if 2 alleles are different, means capital A, small a, these are different alleles, right? Then we call it as heterozygous. Okay, small a is homozygous recessive because it is not dominant. Then capital A is called as homozygous dominant because it dominates, because it is a king. Okay, it dominates the other person, means smaller one, that is a recessive one. So let us find out the frequency of each one of them. The frequency of capital A is P, right? The frequency of A is P. So the multiplying P into P means P into P gets P square. Okay, P square. Okay, then small a is small a. We know that the frequency of small a is Q. Multiplying Q into Q, you get Q square, right? Then capital A into A is 2PQ. Okay, capital A into A, small a is 2PQ. Then from the observations, we can understand this is the expansion of P plus Q the whole square. In the math subject, we have learned the formula, that is a plus b the whole square, that is a square plus 2ab plus b square. Similarly, here by using frequencies, we can form the expansion, that is p plus q the whole square. So it is p square plus 2pq plus q square. So this is the frequency, okay, in the expansion form. Okay, so individual frequencies are represented as P and Q, such as in a diploid, where P and Q represent the frequency of allele capital A and small a. The frequency of capital A is P square, that of small a is Q square, and that of capital A and small a is 2PQ, as I already understood. Okay. Hence, P square plus 2PQ plus Q square is equal to 1, which is the expansion of P plus Q the whole square. As simple as that. So, when the frequency measured is different from that expected, it is indicative of evolutionary change. Okay, what is this? So, when you measure the frequency is different from your assumption or expectation, then it is known as indicative of evolutionary change. Then there is a change in the evolution. Okay, this is what happens. Very simple. So, let's move. Hardy-Weinberg Principle is affected by. So, there are some important disturbances such as gene flow, genetic drift, mutation, genetic recombination, natural selection. These are some important terms that will affect the Hardy-Weinberg principle. So, the frequency of occurrence of the alleles must remain constant. Constant means the total allelic frequency must and should remain 1, right? Constant. So, sometimes what happens due to this disturbance, gene flow, genetic drift, etc., what happens? The frequency of occurrence of alleles does not remain constant. So, let us understand why it happens, how it is affected. Okay. So, Hardy-Weinberg principle is affected by gene flow or gene migration. So, let us understand how it is affected. Okay. Consider this is a population. Okay. This is a population, right? If the gene from this particular population start migrating somewhere else, okay, it start migrating somewhere else, then they start producing there, right? Then they start producing progenies over there. Okay, but it will affect the gene pool. It will basically affect the gene pool. Okay, if some people go out from this population, that will not affect. If the population is very small, then it will not affect. But if large amount of people transfer from this population to here, then what happens? Then it will surely affect the gene flow. Okay, then the frequency of the alleles will not be constant. It will not be constant. It will change. Very simple. The next point, genetic drift or changes occurring by chance. What is genetic drift? So, genetic drift and this gene migration are quite same. They are quite little bit same, but they are different. Okay, at some extent they are same. Okay, genetic drift and gene migrations. Okay, next one is genetic drift, changes occurring by chance. So, genetic drift and gene migration are same up to the particular extent. They are little same. Okay, but up to some extent. But let me tell you the difference. What is the difference? Okay, the very first thing in the gene migration is particular population is migrating to somewhere else, right? The particular population is migrating to somewhere else. Okay, what happens in the genetic drift? Genetic drift is also same, but it can be an accidental event. So there might be injury to a person, or there might be accident has taken place. Then what happens? A particular population transfers to the other place. This is known as genetic drift. This mainly occurs by chance. Okay, some populations transfer from one place to another, might be due to the injury in that place, or might be due to the fire accident has been taken place, or many reasons. There might be the illness to all the people in that place, so they get transferred from one place to other place due to some quite reasons. They are called as genetic drift. So genetic drift and gene migrations are same thing, but up to a little extent. They are not completely same. Okay, very simple. Third one is mutation. So first of all, you should understand what is mutation. So mutation is the large differences occurring suddenly in the population. So let us understand how the mutation occurs. So whenever there are mutations in a gene pool, it will be not equal to 1 constant, right? So we do not need mutations because gene pool will not be remaining constant, so allele frequency will also not be equal to 1. So mutations must not be there because large difference occurs suddenly in the population. This is known as mutation, which is given by Hugo D. Virus, okay, when he was working on the primrose. Next one is genetic recombination. What happens in the genetic recombination? So sometimes during the formation of the gametes, recombinants are formed, right? Gametes formation. Okay, and if it start producing new sequence, then surely it will affect the Heisenberg principle, obviously. So this is really affecting. Okay, so the recombinants must not happen because gene pool will not remain constant. So due to the formation of the genetic recombinants, gene pool will not be constant. Very simple. Then let us understand how natural selection affects the Hardy-Weinberg principle. So we have already talked about the natural selection in our earlier classes, right? So natural selection is that where the sum of the characters are inherited. Some characters are inherited, right, from one generation to another generation, which will surely make particular individual to properly utilize the things which are present in the nature. It will make, obviously, so they are able to survive and are able to produce the new species, right? So they are showing considerable variations, changes, and large amount of mutation in a population. Okay, so which will surely make new species, and this will affect the gene flow, and the gene flow will not remain the constant, and this is what happens. Very simple. Then let's move. This is founder effect, which is very important. Okay, so sometimes the change in allele frequency is so permanent in the new sample of population that they become a different species and the original drifted population becomes the founder. This effect is called as founder effect. So let us consider this is a population, okay, and this population have different kinds of organism. So let us consider A, B, C, D, E are the different kinds of organism present in this population. So what happens? So it will go to different area. So consider what happens. This is a D, right? This is going to a different population. D is going to different population, and then what happens? Then it will surely make many D there. So here it will make many D in this area. It will make many D over there, and it will make new species. And since new species are formed of the D, this is known as founder's effect.